Good afternoon, everyone. Good afternoon, Professor Nares. Good afternoon, Professor Harno.
Yes, good afternoon. Thank you.
How are you, Professor Harno?
Good. Nice. Thank you.
Yeah, thank you for joining with us today, and then thank you for your availability.
Yeah.
Are you in Yogyakarta right now?
I hope in Bandung, but now
Oh, yeah.
in Yogyakarta. Yes, I'm in Yogyakarta, and this is for UGM. This start the first week of after UTS. After.
Oh, yes.
Yes. Mm-hmm.
Yes. All right. I wish I could go to Yogyakarta as well, because Jogja is such a lovely city. Maybe I've been there 3 times.
I see.
Yeah, I should have gone there more often.
Yes, but not every touristic places is open now.
All right.
Yes, part by part, I think.
All right. What about in the campus? Is it also open right now?
Next week we will start with what they call mix between daring and luring.
Yes.
The online. But I think it's almost maybe 50% students still in hometown.
Yes. Yeah. Yes, same also here in Universitas Pendidikan Indonesia. We try to do blended learning.
Yes.
Yeah, quite challenging.
Hello, yeah. Prof. Nares.
Hi, hello.
Hi, Prof. Nares.
Sorry, I was having a little bit of a problem with the Zoom, but now it seems to be fine.
Okay.
Good. Yeah, so thank you also, Professor Nares. We will be starting at 1, so still a few minutes to go.
Yeah, that's true. It's true.
Yeah.
Today I'm at home. Last week I was in the office, but
yes.
Today.
So work from home.
Yeah, that's true. That's true. We are slowly opening up now quite a bit, so the traffic is increasing.
So the normal again, Prof. Nares.
Yeah, that's true. That's true. That's true. And we have a big queue outside the barber shops because people didn't have haircut for many weeks now.
Many weeks or many months.
That's right. That's right. That's right. So
this I got from Pak Bambang Purwono and Tuti.
All right.
Prof. Nares.
Yeah. Hello. Yeah. Thanks.
I said that I will meet with Prof. Nares in UPI.
Okay. I just closed my door, so give me 1 second.
Okay.
Okay, that's better. Good to see you in video, Hannah.
Yeah. Yes.
I'm sorry because I couldn't use the virtual background actually. So yeah, my virtual background is really my white wall.
It's okay.
Can do it on my computer anyway. So yeah, pardon my background.
This is your really background, yeah?
Yes.
Not the virtual.
With the carpet behind me. So yeah, still 5 minutes to go.
Yeah.
And
This Prof. Asep Kadarma also joined with us, the formal rector of UP.
yes, yeah, we actually also invite all teaching team in our department.
Yeah.
Hopefully he can join.
He is my best friend during also in
see.
Yogyakarta during to get S2 and S3, yeah, master and doctor degree.
Yeah.
Prof. Nares, how about the laboratory? All the students is
so from last week we have 50% of the research students are back in the lab. That's the PhD and master students.
Oh yes.
But the undergraduates, they are all online, so even the lab is online for them.
I see. It's almost the same now. Only in my university UGM, there's only 4 research. It is very small number of students.
Yeah, but I think from next week they are saying that we will get more people coming in, so we might be allowed 100% next week.
Yeah.
Because in New South Wales, the vaccination rate is now about 80%. 80% of people are double vaccinated. So that means they can open up even more. And they are going to allow international travel, I think, in December, beginning of December.
Beginning of December. I remember that before pandemic, there is a student from our university to make a research in your laboratory.
Okay. Yeah.
But now it's really close. I hope that next semester maybe we can send student to you.
Okay.
Yeah, 1 more minute while we are waiting for the participants.
No problem.
All right, it's already 1 p.m. in Bandung, and in Nares place, it's now 5. So shall we start right now? Yes. All right. Good afternoon, everyone. Welcome to the webinar series held by Department of Chemistry Education, Universitas Pendidikan Indonesia, or UPI for short. I'm Vidya Vinanuraini from Department of Chemistry Education. UPI will be your moderator for today's webinar. First of all, I would like to say thank you to Professor Nares and then for Professor Harno for joining with us in this afternoon. And also thank you for all participants who make your time attending the webinar, although virtually. I wish all of us the best health amidst the COVID-19 pandemic, because we know that the pandemic is still occurring right now. And second of all, I would like to inform you that today we will discuss about drug discovery and development, and we bring you two speakers. There will be Professor Naresh Kumar from the University of New South Wales, Sydney, and then Professor Harno Dwi Pranowo from Universitas Gadjah Mada, Indonesia. Professor Naresh will talk about the antimicrobial drug development, and Professor Harno will talk about the drug discovery based on the study of the molecular docking. We may all know that drug discovery and development is such a long process. It's not like a fairy tale where you can invent and discover the drug in just one night. It requires a lot of process, from the target identification, hit identification, validation, optimization, up to the toxicological studies and then clinical trials. It's time consuming, and of course, it's expensive. So whenever I took medicine, what I think is, does this medicine or does this drug cure me, or what kind of side effects that this drug have, without thinking too much how this drug invented or discovered. So by this webinar, hopefully we can more understand about the drug and discovery and development itself. And also, I would like to remind you that our webinar is live streaming through the YouTube channel Departemen Pendidikan Kimia UPI. In case you have trouble with your internet connection, so you can join with us via Zoom. You can go to our YouTube channel and you can find the recorded version of this Zoom meeting. And also, after each talk, there will be Q&A session, and you will be allowed to speak if you want to deliver your question directly to the speaker, or you can type or write your questions through the chat box. All right, everyone, I know you are waiting for Professor Naresh and Professor Harno, but I would like to introduce you to our first speaker, Professor Naresh Kumar. He is currently teaching at the School of Chemistry, UNSW, Sydney. He graduated from University of Wollongong, Australia, where he finished his doctoral degree. And he has a strong record in the design and synthesis of bioactive molecules, especially in the development of quorum sensing inhibitors and designing and developing novel small molecular antimicrobial peptide mimics, or SMAMP. And maybe Professor Naresh will talk more about this antimicrobial drug development. And Professor Naresh has also age index of 36. We can see some of the book chapters and publications of Professor Naresh. So yes, these are the most recent journal articles from Professor Naresh about novel seleno and thiourea containing dihydropyryl 2,1 analogs as antibacterial agents. And then Professor Naresh also wrote about nitric oxide donors as antimicrobial agents, and also development of fimbrolized halogenated furanones and their analogs as antimicrobial agents. So a lot of work involving antimicrobial agents. So me personally, really curious about this drug development. So it is with special excitement that I would like to invite Professor Naresh Kumar to deliver your talk in this afternoon session. Please welcome Professor Naresh.
Thank you for your kind introduction. And I'm going to start sharing my screen so that you guys can see my slides and share. Okay. I just decrease that there and maybe go to the and get a laser pointer and laser pointer so you can see me. Okay. Thank you very much for the kind introduction. And I'm going to talk to you today about development of one particular type of antimicrobial agents, which is the short amphiphilic cationic peptidomimetics. And I'll show you what it means when we say amphiphilic, what is cationic, and why these peptidomimetics are important in terms of drug discovery. Just go to the next slide. Just before I start my presentation, I just want to share a group photo. This was taken 2 or 3 years ago, and you can see Vidya just there.
It's me right there.
That's outside our building. And the work I'm going to describe today is mainly done by 2 researchers from my group, Rajesh Kupasamy and Shashidhar. And also Tom also contributed to this project. Veena is another student from Indonesia who did PhD with us, and now she is working as well. And she worked on antimicrobial hydrogels. Shashi is now working in a pharmaceutical company in Perth, and Rajesh is working with us in the University of New South Wales. First of all, you might ask, where is this UNSW in Australia? This is a picture of eastern suburbs of Sydney. And the university is located starting right from this main road over here, going all the way, 3/4 of the way up to here. And there's a big hospital here, which is also part of the university, is called Prince of Wales Hospital. And as you can imagine that we are very close to the beach. And the standard joke in our group is that you can have your lunch, then go for a swim and come back to the lab again. So it's so close. And we are located somewhere here in terms of School of Chemistry. We got a brand new building, which is located right here in the School of Chemistry. Okay, so why do we need antimicrobial development? And as you will know that antibiotics are life-saving drugs, and all of you would have taken one antibiotic or the other at one stage of your life or the other. And some of the very common antibiotics which are prescribed are the penicillin antibiotics. And then if you get a throat infection or a chest infection, the first antibiotics that is prescribed is a penicillin-based antibiotic. Then there is a sulfonamide type antibiotics as well. And also vancomycin is one of the antibiotics which is used as a last line of defense in hospitals when people have serious antibiotic resistance infections. Antibiotics have improved the quality of our life quite a lot. One of the problems with antibiotics is that antibiotics try to kill bacteria, and bacteria know how to survive. And they develop resistance very quickly because bacteria has been on this planet for much longer than we have. And killing bacteria creates this pressure for bacteria to develop resistance. And that's a huge problem. It is predicted that by 2050, more people will die from bacterial infections than cancer. Let me just show you. You can see at the top here, in this timeline, 1930 to 2015, the top shows when the antibiotics were developed. And if you look underneath, it tells you when the resistance was observed. Okay, so the penicillin antibiotics were developed in the 1940s. And by 1946, bacteria were becoming resistant to penicillin already. And the same thing applies to any antibiotic that was discovered, and the resistance developed a few years later. So that's a huge problem because no matter which antibiotic we develop, we get resistance. And that's why many pharmaceutical companies, they don't want to invest into development of new antibiotics because bacteria develop resistance a few years later, and the pharmaceutical companies are not able to get their money back from the investment they do in developing these antibiotics. Because as you know, the development of antibiotics or drugs can cost hundreds of millions of dollars. So there is a real huge challenge for medicinal chemists and the chemists around the world to come up with new ways of targeting bacteria, and how can we get rid of this bacteria which are resistant. Another reason why it is hard to treat or kill bacteria is that the bacteria form what we call biofilms. Bacteria, they come and land on a surface, then they form like a microcolony, and then they form like a mushroom breaks down, and bacteria go in where the places they disperse. And the mushroom structure here, the biofilms here, help bacteria survive. They help bacteria survive antibiotics, because antibiotics cannot penetrate some of these biofilms, and bacteria survive. And up to 80% of human infections, they are related to biofilms. And it's a source of chronic infection, and it protects bacteria in that environment from the host defenses. So that's why it is hard to kill bacteria as well. And a classical example of a biofilm is the biofilm on our teeth. When we get up in the morning, that's an example of a biofilm. And also, with the modern development, the number of biomaterials which are used, they are increasing by the day. And biomaterials are used in a variety of medical devices and applications and implants. A simple example of a medical device is contact lenses. There are 150 million contact lenses wearers in the world. And also there are 150 million vascular devices implanted every year. And there are 3.25 million hip, knee, and fracture implants in the US alone. Although these biomaterials, they increase the quality of our life, but the infections which are associated with these devices is a huge problem. Up to 45% of nosocomial is the hospital-acquired infections. They are related to the devices. There's a huge treatment cost, and it can cost something like US $11 billion every year. And the treatment options are also very limited as well. So what are the alternatives? What can we do in addition to having these antibiotics can we have new ways of killing bacteria? Is there another way we can try to target bacteria? So that's what I'm going to talk about today. And one of the way we've been studying and developing in our group is called antimicrobial peptides. Antimicrobial peptides, short AMP, are short chain of amino acids. So peptides are basically amino acids linked together. And typically there are less than 50 residues. The residues mean 50 amino acids. And they are positively charged. And they're amphiphilic. And I'll show you what we mean by amphiphilic. Amphiphilic means that they have a hydrophobic character. That means they want to run away from water. And they also have a hydrophilic character, which is the positively charged group that is present in these peptides. So this is an example of an antimicrobial peptide. You got hydrophobic groups on the left-hand side here, on the bottom of this structure. And on the top of that, you have these positively charged amino acids. And there are many antimicrobial peptides reported in the literature, and they have activity against bacteria, virus, fungi, and all that. Even our tear fluids, the eye tear fluids, they have antimicrobial peptides. So when a bacteria land in our eye or a virus land in our eye, these antimicrobial peptides can kill them. But their key property is that they're amphiphilic. They have hydrophobic groups as well as positively charged groups. And I was just going to show you where these positive charges and the hydrophobic groups come from. And the hydrophobic groups are typically the amino acids which are water-fearing, and they normally have a non-polar side chain. And an example of a non-polar side chain amino acid is valine, alanine, isoleucine, and so on. They have this alkyl side chain, give them this hydrophobic character. On the other hand, the positive charge in these peptides come from these amino acids, which are polar, and they have cationic side chains like lysine and arginine. So there's a positive charge associated with these amino acids. So these amino acids, they give the peptide the positive charge, while the other amino acids give the peptide the hydrophobic character. So the antimicrobial peptides have both the hydrophobic character and the positive character. Luckily for us, the bacterial membranes, this is an example of a bacterial membrane. It's a typical phospholipid bilayer structure. They are negatively charged. The bacterial membranes are negatively charged, and these peptides, they are positively charged. So the positive charge of the peptide can interact with the negative charge on the bacterial membrane. And when this interaction happens, the membrane structure is destroyed. It can be destroyed by many different mechanisms. And I'm not going to go into all the details here, but what I want to show you is that it can basically form like a hole in the membrane of bacteria. And if the membrane hole is formed, then the bacteria will basically die because all the inside of the cell will ooze out. But luckily also, our membranes, the human membranes, they are neutral. They are not negatively charged. So that means antimicrobial peptides have less effect on our membranes compared to the bacterial membranes. So just to give you one example of the antimicrobial peptide, which we discovered and designed based on copying nature, because nature is the big source of inspiration in drug discovery. And we took two examples. One is called protamine. Protamine is an antimicrobial peptide, which is found in sperms of vertebra. And this peptide here is active against gram-negative bacteria. And you can see they've got lots of R groups and S groups, and R stands for arginine amino acid, and S stands for lysine amino acid, which have the positive charge. So there's a lot of positive charge in this peptide. On the other hand, if you look at the bee here, the toxin in the honey bee, when it stings, contains a peptide called melittin. This is called melittin. And this is the amino acid sequence. All the letters here represent the amino acids which are present in this peptide. And this is the principal toxic compound of the honeybee. It is active against bacteria, but it's also toxic. So what we did was we took part of this protamine and part of this melittin and combined it together to form a new peptide, which we call melamine. It's like a synthetic melamine, synthetic antimicrobial peptide. And if you take that peptide and treat bacteria in solution with this peptide, this is an example of a gram-negative bacteria. And when then gram-negative bacteria is treated with this antimicrobial peptide called melamine, it forms a hole in the membrane of the bacteria. And you can see that from the tunneling electron microscopy image which we have of the bacteria. If you take gram-positive bacteria, Staph aureus here, this is before treatment. And when you treat it with this antimicrobial peptide, you can see that bacteria shrivel up and they sort of die upon treatment with this antimicrobial peptide. These antimicrobial peptides are really nice. They have good activity, but there are certain problems which are associated with antimicrobial peptides. And one of the problems with antimicrobial peptide is that they have in vivo toxicity, like you can't really inject them into the bloodstream, or you can't just take a capsule of antimicrobial peptide because it will get degraded by the enzymes in our stomach and intestine. And also peptides have very high manufacturing costs when we try to make them in the lab. A possible solution to these antimicrobial peptides is to develop these small antimicrobial peptide mimics, SMAMPs we call them in short. So having got this idea from nature that if we can have a positive charge in hydrophobic group within a molecule, maybe we can design a compound which can kill bacteria. So what we did was then we set off to designing peptide mimics, which are antimicrobial based on this idea. But before we did that, we thought, let's check what is there in the literature. And the literature also have examples of compounds, examples of these antimicrobial peptide mimics, which have a core structure. The scaffold in the middle is made up of these 4 rings. But on one side of the molecule, you have this blue circle, which represents the hydrophobic groups. And on the other side, we have this red group, red circle or red oval here, which represents the positive charge within the molecule. So there's a hydrophobic group, there's a positive group. Another class which has this backbone in the middle, and then you have the hydrophobic groups at the bottom. These are tertiary butyl groups, and these are the positively charged groups on the top. And this molecule here has also similar characters. You've got a hydrophobic group on this side, which is your tertiary butyl groups. And also you have your positively charged groups on the other side. And this molecule is currently going clinical trials for treatment of bacterial infections. So in my group, we thought, can we come up with new scaffolds? Can we come up with new structures which will allow us to develop these antimicrobial peptide mimics? So we worked on 3 different scaffolds, and I'm going to briefly talk about all these 3. One is called as a phenyl glyoxamide scaffold. Other one is called a biphenyl. And the third one is just a biphenyl, just a phenylamide. And if you want to more details about these scaffolds, you can read these papers and they have that information in them. So what do we mean by this glyoxamide scaffold? What is the difference between a normal amide and a glyoxamide? You would have all come across an amide bond before, and you can see that amide bond is basically a CONH group. And CONH group is present in all peptides and proteins because the amino acids are linked together by these amide bonds in these peptides and proteins. So what we decided instead was that instead of having just simply an amide group here, can we use a glyoxamide group? A glyoxamide group is when you have a CONH group. And the reason for introducing this glyoxamide group is that these glyoxamides are quite stable. They cannot be simply chewed up by the enzymes, and they also tend to be non-cytotoxic as well. So this was the reason for choosing this phenyl glyoxamide scaffold. And I'll talk about the biphenyl later. Biphenyl is when 2 benzene rings are joined together. That system is called as a biphenyl. And this is just a simple amide-based structure. So let's just talk a little bit about phenyl glyoxamides, because that's the first scaffold we started off when we started off this project. So the question then arises is, how are we going to form this glyoxamide scaffold? How are we going to introduce that functionality into the molecule? The design is very simple. The design is you have a positively charged group on this side, and you have a hydrophobic group on the bottom. So R1 is a hydrophobic group, and it has this glyoxamide group present in it. And one way you can get that glyoxamide group is by using a very simple naturally occurring molecule called ecitone. And ecitone is a very common chemical. And the closest thing to ecitone you might have come across is the indigo dye, which is a dimer of ecitone type compounds. And this compound occurs in nature. So what we wanted to do was that if we can ring open this 5-membered ring here, then we will generate a glyoxamide based structure. And what we learned when we started this project was that if we take an ecitone molecule, and then you put a hydrophobic group onto that ecitone nitrogen by a CO group, forming an acyl group. We call that an acyl group on the nitrogen. This carbon number 2 here, the CO number 2 here, becomes highly reactive, and it can ring open quite easily with an amine to form a glyoxamide group. And when we use a dimethylamino compound, we can generate this dimethylamine here, and we can convert this nitrogen into a positive charge quite easily by simply forming a salt, or you can simply alkylate it to get the quaternary ammonium salt. And the hydrophobic group we saw, which works the best, is a naphthyl group rather than a long alkyl chain, which is present in other peptide mimics. So having made the first series of compounds, we decided to test whether these compounds have any biological activity. So this is some of the examples of the compound which we tested for activity. We had the ammonium salts and the corresponding quaternary salts. So what I got in blue here is MIC. MI stands for minimum inhibitory concentration. And we tested these compounds against gram-positive bacteria called Staph aureus. And this compound does have some activity, like 16 microgram per mil against the gram-positive bacteria. But when we tested the quaternary ammonium salts, we found that they had less activity compared to just the salts of these compounds. But what we also found was that when we checked the cytotoxicity of these compounds, so the red values here, they are the cytotoxicity against the mammalian cells. And we found that these compounds were quite cytotoxic, except the quaternary ammonium salts were less cytotoxic, but the salts of these amines, tertiary amines, were quite toxic. So we really started to think, how are we going to reduce the cytotoxicity of these compounds? Because a drug is useless if it has a lot of cytotoxicity. So we decided that from the amine compound, we will make the corresponding guanidine compounds and then check whether these guanidinium compounds are less toxic or more potent or less potent compared to the quaternary ammonium groups. So we wanted to see if we can convert this amine group here into a guanidine type group. And we came up with these three series of molecules which we thought we might make: series 1, series 2, series 3. I'm just going to show you the synthesis of series 1 and series 3, and then show you some activity, whether these compounds are active or not against the bacterial cells. The synthesis of these compounds is very simple, and I'm not going to go into too much details of the reactions we did, but simply to show you that if you take that naturally occurring acetone molecule and introduce this hydrophobic group onto the nitrogen here by an acylation reaction, then you can ring open this carbonyl group here, the 5-member ring here, by attack onto this carbonyl to give us the glyoxamide group. And this protecting group here, BOC, is a protecting group that can be removed to get a free amine. And then the free amine can be converted into a guanidinium group quite easily by standard set of reactions. So this is to form the mono guanidine compound with some positive charge, okay? And then to introduce two guanidine groups, we use a similar chemistry. We take the ester, we hydrolyze the ester to the acid, then we couple it with a second molecule, and then we deprotect that BOC again. And the two amine groups which we have then are converted to the corresponding guanidine groups. So the chemistry, once is standardized, is a matter of routine, is a matter of doing coupling reactions, deprotecting, adding the other group to convert these to guanidinium compounds. So we've got series 1 compound and series 3 compounds. And then we decided to test these compounds as well to see what sort of activity they might have. And we found that some of the compounds here turned out to be quite active. When we tested the activity of these compounds here against gram-positive bacteria, they turned out to be at 10 microgram per mil, the minimum inhibitory concentration. And we found that these compounds were also active against gram-negative bacteria as well. And the purple value here is the value against E. coli. E. coli is a gram-negative bacteria. And when people have urinary tract infections, E. coli is present in many of those infections. So these compounds turned out to be quite active in terms of their activity. Then we also tested whether these compounds could inhibit biofilm formation. You may recall that we talked about that 80% of human infections are biofilm related. So can we stop or can we kill the bacteria which are present in biofilms? Because then obviously you will be able to use them in real life infections. So we found that these compounds here, particularly compound 65, 66, and 37, they reduce they disrupt the biofilm by quite a bit percent in these assays. This is measured by using a fluorescent dye, and these images are taken by confocal microscopy. The green here indicates bacteria growing happily into biofilms, and the red here means bacteria are dead. So you can see that in the treated biofilm, bacteria are pretty much dead, while the normal not treated biofilm bacteria are flourishing. So that means the bacteria, the compounds are active against bacterial biofilm as well. The last thing to test was, do these compounds have cytotoxicity? Because cytotoxicity obviously is very, very important. And you can see here for the compound 35 and 36, the minimum inhibitory concentration required to kill bacteria or inhibit 50% of to inhibit bacterial growth was only 6 microgram per mil, while the compound is cytotoxic at 174 and 224 micromolar. So that means these compounds have a big therapeutic window. So therapeutic window is typically measured by looking at IC50 against a mammalian cell, divided by the active concentration, which is 8 in that case. And you can see there's a big margin between the activity and the cytotoxicity. So we designed and tested this first series of compounds, which turned out to be quite active. And some of these compounds are now being evaluated for applications in industry in terms of treatment of bacterial infection. Okay, the second scaffold I'm going to talk very quickly is a biphenyl scaffold. So the idea here is exactly the same, but what we have is a structure which is a biphenyl structure. And on one side of that biphenyl structure, we're going to introduce the cationic groups, positively charged groups. And on the other side of the scaffold, we're going to introduce this hydrophobic group in that structure. And the types of cationic groups we're going to use are the cationic groups which are indicated here on this slide. And you can see that these are the cationic groups we're going to use. Okay. And again, the preparation of these compounds are going to give you a very simple overview how this scaffold is made. We just take two components together, one which has a bromo group and one which has a boronic acid. And there's a very famous reaction called Suzuki-Miura coupling reaction. And that reaction can be used to couple these two benzene rings together to form this biphenyl structure. The nitro group is then reduced to the amino group, and the ester group is there as a protected group. So what we can do is then use this amine group here to introduce hydrophobic groups on this side of the molecule. And then we can use the ester group later to actually introduce the cationic groups on the other side. So we make the key compound which has the amine functionality and the ester functionality. Then we couple it with an amino acid to introduce a hydrophobic amino acid here. And then we can couple another different amino acid there if we like, to make like a two amino acids over here, where X is tryptophan and Y is phenylalanine. And we can hydrolyze this ester to get the acid. And then the acid can be coupled with various amines that are present, which I showed you in the earlier slide. So we got quite a few different amines we can introduce at that position. So this is just basically the continuation, showing you that the amine can be coupled with the carboxylic acid quite easily. And that allow us to make a huge series of compounds. We can make a library of these compounds, and the library of the compounds help us to do a structure function analysis to see which groups are important for activity and which groups are not important for activity. And just going to show you a couple of examples to show how we can exploit this library to select compounds which are really, really active. And if you just compare these two structures here, which we made, there's a biphenyl, there's tryptophan, there's phenylalanine, same here, biphenyl, tryptophan, phenylalanine. But on the left side of the molecule, you have one amine functionality and one guanidine functionality. While here, you have two guanidine groups. And you can see that the compound with guanidine groups here is more active compared to the compound against which has one amine group. So that means by manipulating the structure of our molecule, we can design much better, much potent compounds. And the biphenyl structure here is very important for activity. If you remove this biphenyl functionality within your structure, you find that the activity is all lost. So there's no activity associated with just the groups which are present in the molecule, but the biphenyl helps the molecule to insert into membranes. And that's why the biphenyl functionality is very, very important. How do we know whether the compound is interfering with the bacterial membranes? We've been telling that the compound interferes with the bacterial membrane. What is the proof? The proof comes from an experiment which we do with bacteria, where we treat the bacteria with a dye, and this dye is called as a cyanine dye. So this dye absorbs into bacteria through their membranes. And if the membrane is disrupted, then the dye will be released out. And we can measure the amount of dye that is produced against time when we treat bacteria with these compounds. More dye is released means more bacterial membrane is compromised. And we could show that with our compounds, we can disrupt the bacterial membranes. So that gives us the evidence that it is acting via the disruption of bacterial membrane. Just to show you as a cartoon representation that how this assay works is that this is your bacterial membrane. And when you treat it with this dye, the red circle here, the red dye will go inside the cell. And when you treat the cell membrane with the antimicrobial compound, the dye is then released from the cell membrane because the membrane is distorted or destroyed and the dye comes out. When the dye comes out, we can measure that dye using fluorescence spectrometer, and we know that the membrane is disrupted. And that's how we do that assay to work out whether the dye is released. Just very quickly, the final scaffold which I'm going to talk about is the phenyl glyoxamide scaffold. Not going to spend a lot of time, going to go through that fairly quickly because it's very similar to the scaffolds I described earlier. The only difference here is the key molecule which we use to make this phenylamide scaffold here is using this molecule here called acetoic anhydride. To make the phenyl glyoxamides, we used acetone, but to make the phenylamide scaffold, we use acetoic anhydride. And again, this compound is readily available. Again, very cheap commercial starting material to make our antimicrobial peptide mimics. The chemistry is again very simple, but very, very versatile. You can ring open this acetoic anhydride as I described before to generate a compound which has an amine functionality and a carboxyl functionality. And this acts as our key starting material, amino functionality and a carboxyl functionality. And the carboxyl functionality is coming from the tryptophan amino acid. And again, on the amine functionality here, we can introduce our hydrophobic group. And on the carboxyl functionality here, we can introduce our quaternary group. And the quaternary group are introduced by simply a coupling reaction. And let's not go into the details of these reactions because you can look at them in your own time. But the key is that you have the carboxyl functionality here, and the carboxyl functionality can then be used to introduce either an amine group or a guanidine group into that structure by a simple sequence of reactions, coupling, deprotecting, and then getting the target molecule. And again, you can vary this functionality X to whatever you like, giving you again a library of compounds which you can exploit in terms of structure function and come up with more active molecules. We can also make the quaternary ammonium compounds using the same chemistry I described earlier, coupling the acid with the amine. And once you have amine, you can either form a salt of the amine, or you can form a quaternary iodide salt or a chloride salt with that compound. What about the activity of these compounds? Are they active? This is the advantage of having a library. So you got fluoro, chloro, bromo salts, you got bromo, chloro, fluoro, quaternary salts. So you can compare the effect of changing one group on the activity of your compound. Is it increasing activity? Is it decreasing activity? You can learn by doing these testing. And that's why medicinal chemistry, when there's a drug development happening, there are thousands of compounds made. And out of thousands, only 1 or 2 are selected which go forward because they meet all the criteria. Just to give you another example, we are simply comparing in these 4 structures at the top and the 4 structures at the bottom. The only difference between the top structure and the bottom structure is this has an amine group and this has a guanidine group in all structures. Amine, guanidine. And you can see that in all cases, the guanidine compounds are performing better than the amine compounds. This compound, lower value here, means more active compound. So that helps us to design, to select groups which gives the molecules the best activity we want. And obviously the cytotoxicity is very important, because if the compound is cytotoxic, there is no point in using that compound any further. And what we found was that some of our very active compounds like 9B, this compound here, for example, is not cytotoxic at all. MRC5 cells is human cells, and it is no cytotoxicity at 100 micromolar or 50 micromolar. The cells grow happily. Same with 9D. There's very little cytotoxicity associated with this compound, but these compounds are highly active. So that means these are our lead compounds. These are our compounds which we are now testing against applications where bacterial infection is a problem, or where bacteria are a problem. And some of these compounds are being added into antibacterial formulations. And even one of these compounds is also active against virus, and it is being trialed in an antiviral formulation at the moment in collaboration with a company in Sydney. We can again do dye release assays to show that these compounds disrupt disrupt the membranes. And that is again done using the same assay I described earlier, where the dye is incorporated into the bacterial membrane. And when we treat it with our compound, we can see how much dye is released. And increased dye typically is associated with increased death in bacteria. So that is indicating to us that disruption of bacterial membrane causes the killing of bacteria, that these compounds are bactericidal. And also these compounds are active against biofilm as well, because biofilm is a key survival mechanism for many bacteria. And we can disrupt biofilms. For example, in this case, 9D, our lead compound, can disrupt Staph aureus biofilms by 83%. So that means it's really very active compound and treat infections or treat where Staph aureus is a big problem. I think that brings me to the end of my talk. So what I have done today is given you a new direction or new thinking in terms of how to tackle bacteria and what we are doing, or what is the current thinking about developing these peptide mimics as antibacterial agents. Because as I mentioned in the beginning, bacterial resistance is becoming a huge problem, and there's a need to develop new ways of targeting bacteria. And one way to target bacteria is taking a lead from nature, because nature has produced all these antimicrobial peptides. And what we are trying to do is to mimic that nature in the lab so that we can design small, simple molecules which can allow us to tackle bacteria in the years to come. I really want to thank my group and a lot of the PhD students who I showed you in the beginning of the slide. They have become doctors. Professor Ventis Rianti, she came from Solo, Indonesia, and she also worked on the peptide mimic project. And I mentioned Shashidhar and Rajesh before. And then here are some other researchers within my group who contributed. And I'm really thankful to the funding from the Australian government, Australian Research Council, and the National Health and Medical Research Council. And I'm really thank you for the patience and listening to my presentation. Okay, thank you.
All right, thank you very much, Professor Naresh, for such an insightful lectures in this afternoon. Actually, it's like a gloomy afternoon here in Bandung, in my place.
Is it raining?
Yeah.
Is it?
Yeah, I think it comes to rain. Yeah, shortly.
All right. Okay.
All right. All right. So participants, dear participants, I would like to invite you if you have any questions regarding Professor Naresh's talk. Professor Naresh just talked about the antimicrobial peptide mimics, and it can kill bacteria via disruption of bacterial membrane, and also disrupt the biofilm. So if you have any questions, you can raise your hands if you want to directly ask to Professor Naresh. Right, from Fini. Fini, would you like to ask directly to Professor Naresh?
Like to ask the special requirements function will introduce anti-competitive or just general or any cationic groups. Okay, so there is a question I'm reading in the chat. So the question is that are there any special requirements for the functional groups we introduce to our antimicrobial compounds, or just general or any cationic group or hydrophobic will just do? I will answer the hydrophobic group first, because we investigated quite a variety of hydrophobic groups in our study. We looked at the alkyl chains, we looked at the benzene ring, we looked at the naphthyl group. And in our case, we found that the naphthyl group was the best. And we have also made some different compounds instead of amide, which have sulfonamide groups. And in the sulfonamide groups, we found that the alkyl chains work the best. So it's a matter of trying out with your compound. Try with the simple alkyl chain. If the alkyl chain doesn't work, then you can start thinking about more aromatic hydrophobic groups, and that's useful. And in terms of the cationic groups, we found that the guanidinium groups seem to be the best, because the tertiary amine groups or the quaternary amine groups, we found that they had quite a bit of cytotoxicity associated with them. But once we converted them to the guanidine group, the cytotoxicity was much less. Other people in the literature, they have used even heterocyclic rings, which have positive charge, like imidazole and the like. And other people have even looked at phosphonium groups, which have a positive charge as well. But from our experience, the guanidine group seems to be the best in terms of activity versus cytotoxicity. So I would suggest that you try the guanidinium groups first, and also can try just simple amine group to see what you get. So it's a trial and error process. You just try different groups. You can also do modeling. Sorry, I didn't mention, you can actually model bacterial membranes with your compound. And modeling can also tell you how your compound is interacting with the bacterial membrane. And there are some papers who even describe that from modeling, you can predict how quickly the compound will go inside the membrane in terms of bacterial membrane. So even modeling can help you a little bit in terms of designing as well.
All right. Thank you, Professor Naresh, and thank you, Fini, for your questions. Okay, participants, if you still have any questions about the antimicrobial or similar questions with Fini or other questions, please ask to Professor Naresh. Yeah, actually, I'm curious about the bulkiness of the structure of the AMP mimics. I mean, the bulkier the better, or how does it affect the
that's a very good question. That's a very good question, because you can also imagine that these compounds have hydrophobic groups and cationic groups, and the hydrophobic groups can come together to self-assemble. Like, for example, if you take oil and fat, they have long alkyl chains, and they can assemble as well into structures. And the same thing can happen with the antimicrobial peptide mimics as well, is that when you introduce certain groups in them, they can assemble very well. And when they assemble, they can even form gels. And as you will know, Vidya, that Veena, who did her PhD on this project, she formed hydrogels. And the hydrogels are fantastic in terms of applications, and those hydrogels are antimicrobial. And the hydrogels are formed by this assembly process, right? But coming back to your question, if the molecule becomes very bulky, obviously, then you raise the question whether it can be used as a drug or not. Because the drug, in my last lecture, you might have heard that I mentioned about Lipinski's rule, that the molecular weight should be less than 500 and number of hydrogen bond donors and acceptor groups. So what we are doing with these compounds at the moment is we are looking for external applications. External application, I mean wound care application. Like if somebody has a wound on their leg which is highly infected, you are not putting the compound into the bloodstream. You're just treating the wound outside with your peptide mimic. So the size there is not important as long as the compound is active. You can just dissolve in a formulation and apply. Other application we are looking at is, as I mentioned, disinfectant formulations, where again you are applying it to a surface. You can apply it to a handle. You can apply it to anything you like, or on your hands as an antimicrobial compound. You can also use these compounds to treat gut infections, where the infection is in the stomach or in the intestine, because then you don't have to absorb the compound into the bloodstream. You're just curing the infection in the stomach. So these compounds can be used for those kinds of applications. But if you want to use them for other applications, you probably will have to come up with a delivery system which will protect the compound when you deliver it inside the body, and then it reaches the target, and then it can be used in like a liposome, or like an emulsion, or like a nanoparticle of some sort, where it could be given in the body itself.
Right. So yeah, it can be used as topical. Not we do not intake it orally, but we can use the compounds for.
But having said that, the antimicrobial peptides, there's an antimicrobial peptide called colistin. Colistin is again used in the infections which are very, very severe, and that is given as an injection.
Right.
So colistin is an example of a peptide, but it is given as an injection, and that is still active, right? So in severe infection cases, you can use Gramicidin is another antimicrobial peptide, which is antibiotic, which is used as well. So they can be used. It's just a matter of the structure, the type of structure you have, I think.
All right. So we still have, thank you, Professor Naresh. We still have 5 minutes before we go to the second lecture. Okay, Vinny, thank you for your explanation. All right, dear participant, we still have time if you have any question.
One thing maybe I can tell everybody is why interrupting the bacterial membranes wouldn't cause resistance, because with antibiotics we get resistance, with the antimicrobial peptides, we are saying there'll be less resistance or no resistance. The reason is that antibiotics, they target one particular enzyme in the body. They target one particular receptor in the body, in the bacterial system. So if there's only one receptor or enzyme is being targeted, then bacteria can just change the receptor, or they can change the enzyme or modify the structure of their enzyme, and the antibiotic becomes useless. And bacteria can also produce chemicals which can break the ring within the antibiotic, and that's where they become resistant. But the antimicrobial peptides and mimics, they destroy the whole membrane. And there is no simple way for bacteria to restructure their membrane. They cannot just redesign the whole membrane. So that's why if the membrane is disrupted, bacteria are killed. And there is no easy way for them to change their membrane. And that's why there's less development of resistance. And antimicrobial peptides have been there for a very long time. All humans, mammals, they all have antimicrobial peptides as a first line of defense against bacteria and viruses. And there is still very little resistance to the antimicrobial peptides compared to antibiotics.
All right, so can it be effective for superbugs?
Yes, I think that's why there is a lot of research going on in antimicrobial peptides and mimics, particularly targeting superbugs. So the two bacteria I described today was Pseudomonas aeruginosa is one, E. coli is another one, and Staph aureus is another one. So you have a resistant strain of Staph aureus called MRSA, which is methicillin-resistant Staph aureus. This is also called that golden staph. So once we test the compounds against bacterial strains, they're active. Then we also test them against resistant strains as well, and also, the so-called resistant superbugs and antimicrobial peptides can still show activity. And also, we also try combination of peptides with other compounds as well, so that we can have two compounds present, two different mechanism of action compounds present in the same mixture. And that can also target superbugs as well. Maybe one drug cannot kill it, but you can combine it with an antimicrobial peptide and a drug, and then you can tackle it.
Right. So yeah, the good news.
Yes.
All right. Question from Adam.
Okay, Adam, is there any specific purposeful meaning of using benzimidazole ligand in your produced drug? Thank you. I mean, you can use imidazole to form a quaternary group, because you can imagine if you take your imidazole and then alkylate the both nitrogens, you have a positive charge, right? And you can do that with benzimidazole as well. And people have tried imidazole quite a bit. Not as much benzimidazole, but it's worth a try to see if you can incorporate that positive charge by using a benzimidazole ring. But I would try an imidazole first, which is a 5-member ring with 2 nitrogens, and then see what you get. And even you can try pyridinium. If you take pyridine and form a pyridinium salt, that also gives you a positive charge. So it's worth trying it, yeah.
All right. Thank you, Professor Naresh. And thank you also, Adam, for your question. It's now 2 p.m. So I think we come to an end for Professor Naresh's talk. Thank you very much. And on behalf of the Department of Chemistry Education, we would like to gratefully thank to Professor Naresh for your availability and time to deliver the talk in this afternoon's webinar. So please we would like to once again thank you, Professor Naresh. Thank you for your contribution to our webinar.
Thank you for the opportunity. I really appreciate talking to you and giving me the opportunity to connect with you guys again, and it's good to see some familiar faces.
Yeah. Thank you. So, is there anyone to deliver or talk about your presentation, maybe, or concluding remarks?
Sorry, are you asking me?
Yeah, Professor Nares, if you, for
okay. Yeah. Look, I really really appreciated and enjoyed giving a seminar. We are in a very different world at the moment. I would have loved to give this talk in Bandung itself, but unfortunately it's hard to travel, and this is a good platform to have lots of people in their own places to listen to the presentation. And I think this webinar is a very good idea to have a lot of people together and listen to presentations. And really, I'm impressed with the interest which is there in the webinar. I was looking at number of participants. There's about 146 people present in here, and which is really good. So that shows that there is a keen interest in education and in learning about this drug development in your university and in Indonesia. So I really enjoyed giving the talk, and I'll be happy to answer questions if you have later on. You can send me an email, or you can contact me some other time when I give other lectures. So thank you very much, and I really appreciate your attention.
All right, thank you, Professor Nares. And now it's time for us to take picture maybe together. Yeah, before we jump to the second lecture from Professor Harno. So please do turn on your camera if you don't have any trouble with your internet connection. Right. Good afternoon, Bapak Hayat Solihin.
Hello, good afternoon. It's a nice, interesting lecture.
Thanks, Solihin.
All right, is everyone ready for taking picture? We have 6. All right, please put your smile. Okay, 1, 2, 3. Okay, the second one. 1, 2, 3. Right, third. Okay, the fourth. Right. Okay, last one. Right, that's it. Yeah, thank you once again, Professor Nares, for your time. Hope, we still meet you. Yeah, we will still meet you in the next 2 weeks in November, because Professor Nares will still deliver the lecture at 4th of November, if I'm not mistaken. Yeah. All right. Thank you, Professor Nares. And right, everyone, we will start our second lecture from Professor Harno, but as per usual, I would like to introduce you to Professor Dr. Harno Dwi Pranowo. He's currently teaching at the Department of Chemistry, Universitas Gadjah Mada, Indonesia. So he graduated from the University of Innsbruck, Austria. Hopefully I'm not mistakenly saying the university name. Where he got his doctoral degree, and his field of expertise is computational chemistry and then molecular modeling and drug design. That's why in this webinar, Professor Dr. Harno will talk more about the drug design based on the study of the molecular docking. Professor Harno has a Scopus H index of 8, and he also wrote some books. There are 3 chemistry books and 5 non-chemistry books, and Professor Harno published articles with the total citation of 233. And these are the recent publication from Professor Harno, design of new chlorochalcone derivatives as potential breast anti-cancer compound based on QSAR analysis and molecular docking study. Also, cobalt 3+ and iridium 3+ in pure liquid ammonia. The structure and dynamics from ab initio quantum mechanical charge field molecular dynamics. And Professor Harno is also a senior president of Indonesian Chemical Society. He is also president of Indonesian Computational Society. And he was also president of Indonesian Chemical Society back then in 2019. He was also head of UGM, Universitas Gadjah Mada Publishing and Publication Agency. So those are to name a view of management experience of Professor Harno. All right, without further ado, I would like to invite Professor Harno Dwi Pranowo to deliver the talk about the study of molecular docking regarding the drug design and development. Please welcome Professor Harno.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Okay, I will give the lecture about molecular modeling in general, and I've been sharing my okay. Okay. Yes. My presentation is okay. You can see my presentation?
Yes. Yes.
Okay.
It's perfect.
Okay. Thank you. So drug discovery based on study of molecular docking. If Professor Nares has already give the information about how to synthesize and how to make that new molecule is active as a give us the bioinformations about some molecule, some new drug, and in this occasion I will give you the information about the molecular docking. One of the technique you have, one of the method to do the drug discovery, especially on drug based on the computational chemistry. Okay. So if we talk about drug design and this discovery, that if we have a drug, so that we need, if the drug, we can actually got from natural sources. We have the natural product that we can, nature will produce the compound that active for some purpose, I mean, as a drug. But of course, if natural resources, it is a very limited number, so that not enough for our need. So that that's why we have synthesis sources. We should get the structure of the compound, testing for the ability to have active, for some as a drug. But of course, we need the synthesis sources to get the big number of the compound that need for people. For example, we have the antivirus for our pandemic, so that we need a lot of drug for this. Because of the synthesis, we have also the target. The study of the target of compound, drug compounds. So it means that we talk about the protein receptor. So that the experiments of the X-ray diffraction or NMR give us possibility to make the database, protein database, protein data bank, to introduce that there is a target molecule, target of as a protein target, and then there is also the compound as a drug that interact with target molecule. So that's why we drug design and discovery almost what we say multi we have the mathematics, of course, we have a medicinal chemistry, multidiscipline is inside the our drug design and discovery. Okay, so if we talk about the medicinal chemistry, it means that this we talk about the synthesis, because the science that deal with discovery or design new therapeutic agents and their development into useful medicine. So normally we have a synthesis, involved the synthesis like Professor Naris talked before. And we have also structure-activity relationship, how the structure of the compound give the relation with the activity of compounds. We talk about amide, we talk about the carboxylic acid and so on, but connected to the activity of the structure as a drug. And the third one is receptor interaction. Mentioned before that for molecular docking, for take account the interaction between drug and protein receptor. So we have some not difficulty, but sometimes it's difficult to make the update of this. But now is I think it's PDB protein data bank is in October this month. I think 50 years for database. So means a lot of data of structure of protein, and also drug is inside and free for download. And then also mentioned before Prof. Naris is if we talk about medicinal chemistry, we talk about the absorption, distribution, metabolism, excretion, and also toxicology. So it means again, this is multidiscipline approach for people working in medicinal chemistry. If we talk about drug discovery, this is a long. We need a long time. Start from identify the diseases, isolated the protein, and some chemistry. The combinatorial chemistry, for example, chemistry discipline with the high throughput screening, now is high throughput virtual screening. It is also needed for how to make a decision, maybe choose 2 or 3 molecules from the database, or big database. There's a need for starting value for try to make calculation in molecular docking or QSAR. And we have also the molecular modeling. I think we can use it for not only for molecular design or drug design, but all chemical, all needed for the know about the chemical system. Of course, it is not only the chemistry here, but we try to find some calculation that using the computational chemistry to find information, useful information for drug design. If we talk about the molecular docking, actually we talk about the intermolecular interaction in a protein system. We talk about the supramolecular chemistry. I like very much to describe the supramolecular chemistry, not because of the big molecule, but we discuss about the interaction one molecule and another. So that's why we need interactions, okay, polar, hydrogen bonding, and so on. Yeah. So because, for example, if of course we need the information, knowledge about the structure of protein. Yeah. The structure of a primary, secondary, and tertiary structure of protein, so that we can try to make the calculation or make the imaging, yeah, that the interaction of protein and compound, that drug compound is in this very big molecule like protein. Yeah, this bonding in protein, we can say that molecular hydrogen bonding is the very important technique how to understand how the bonding in protein working. Of course, there is a van der Waals interaction, charge-charge interaction, and so on. Especially if you have a drug that says like a salt and so on. Yeah, a lot of interaction. But of course, we can explain the interaction using, for example, organic, physical organic chemistry means. Yeah. Again, in computational chemistry, this is very briefly, I get information that there is a CAD, means a computer-assisted drug design, that give us the possibility to make the decision. For example, like during the if we have a lot of data, like Prof. Nare says, we can use the QSAR, quantitative structure and activity relationship, because we have many compounds that are almost similar structure. Then we have also the activity, so that we can make the calculation based on quantitative structure and activity relationship. Yeah, so that finally we have the lead compounds from this part. Other part is like in our pandemic, there is no antivirus for COVID-19, but we have a lot of compounds, yeah, in the database, compound that have the activity as antivirus, but not for molecular for pandemic of COVID-19, so that we can use this opportunity to running the molecular docking calculations, yeah, to find the possibility to find the new compounds that give us antivirus for COVID-19, for example, yeah, with this method. Yeah. And today we will talk more about the molecular docking to find the new compound that has the high activity of the for molecular docking, from molecular docking calculations. It is our topic today. Again, that if we talk about the method to find new compounds, new drug, yeah. So we have the SBDD, structure-based drug design, means that we have the protein receptor, the data of protein receptor, and then we have also the structure of the compound, for example, antioxidant or antivirus. Yeah? Of course, we can find in protein data bank. Yeah? But the new or not new, another method is what we call LBDD. If we have only the structure of compounds as a drug, several, maybe 10 or more than 10 compounds, so we can use this QSAR, for example, or now with QSAR 3D, we possible to use it. And then we can find the possibility to recommend it, recommended new compound that maybe synthesizes and then check for the activity. Okay, sometimes we have the lucky because data, a lot of data from activity and structure of the compounds, so that we can use this LBDD, ligand-based drug design, but also we have the structure of the structure of a protein receptor and also structure of the drug compound. If we have two data, LBDD and SPDD, can running together, so that it is very good condition. But sometimes we have only SPDD or LBDD only. Oops, sorry. Okay. Drug design successes, I think a lot of data. This is just example. Antibiotic using the QSAR as 1983 and so on. This is of course because start from the calculation, QSAR or molecular docking, and then of course there is another difficulties, not only in modeling, but also in synthesis. For example, now in our laboratory, in organic chemistry laboratory in chemistry department UGM, normally for S3 or doctorate degree, the first year working in this program is running the computational chemistry calculations, doing docking or QSAR, and we have recommendations of some compounds, and then the second and the third year, we are working with synthesis and also for the activity. So it means that the people who want to make the synthesis give the information from molecular docking or QSAR studies products. Okay. What is protein ligand docking actually? Definition is computational predict the structure of protein ligand complexes from the conformation and orientation. The people here is very good make the definition that is not only conformation but also the orientation of molecule in the interaction of with receptor, protein receptor. That's why I put here supramolecular chemistry, because we cannot give the information about how to running how to make to designing the computer result of the calculation without known about the supramolecular chemistry. Okay, molecular docking is running is almost the same. First, it's check for the receptor protein. We find a PDB file, and then we have the ligand. Of course, this is for synthesis. We have a lot of data for elucidation structure and so on. After that, we go to the software of molecular docking running. Maybe it depend on the big or small molecule as a drug, maybe 1 month, maybe 2 weeks, and so on. And the analysis is the same. First is the interaction residue, how the residue of the protein will interact with compounds, drug compounds, and the type of interaction And finally, we have the energy produced after docking. So this is the three of point interest of the molecular docking calculations. So what is what we call, this is one of the example, RCSB protein data bank. I always say this RSCB, but nevertheless RCSB, yeah, Research Collaboratory for Structural Bioinformatics. This is, as I say, the October molecule of man. You can find here, and you can download for free all the structure of protein or small molecule also here. And you can find not only the structure, but information about the research. Yeah. After you can find here, we go to software molecular docking. This is Jasmine main software. A lot of another software to find how the computer can help us to make the calculation. One of the software is Autodoc, yeah, and then we can find running the molecular docking calculations using this software. So I will give you the example. Yeah, example. This is from our team, the design, interaction, and simulation of molecular dynamic of quinazoline derivative compounds as an inhibitor of epidermal growth factor receptor, or IGFR. This is the protein IGFR, and this is collected from the RSCSB protein data bank. Yeah, and there is three molecules. Yeah, what we call small molecule tyrosine kinase inhibitor. This is quinazoline, and these two is the erlotinib and afatinib. This is just example that this is actually the same quinazoline here, and put some substituent. Yeah, so this is the first and the second generator. So from this information, we can find this information to make the calculation using molecular dynamic, molecular docking molecular. Yeah. Before using this calculation, I already said that if we have lucky that we have the data for running the QSAR, but also for molecular docking. Here we, for example, if you run in the QSAR quantitative structure activity relationship, means that we have a lot of structure, a lot, and then we can find the mathematical model, mathematical model correlated about the activity of the compounds, yeah, and what they call the characters, character of the compounds. Yeah? So it means that there is the correlation between structure and activity. So this is descriptor of this. Okay. And our students, our group, this has a lot of data. I think at the moment they have 25 compounds at the time. So that the best QSAR equation use electronic and molecular descriptor data. Means that there is not only molecular descriptors like mass, log P, but also the what is called every atoms in the molecule has for example carbon and nitrogen. We can find the what is a quantity of the charts here, or charts every atoms. Yeah, and this is analysis of HOMO, And the volume, log P, and mass means that descriptors, a molecular descriptor. But we have also the electronic descriptor here. And from the QSAR study with recommendations, some compounds, that R1, R2, and R3, R1, R2, R3 is we need the short chain alkyl substituents, for example, to make the new compound that the activity of compound is increased. From this recommendation of QSAR, we can make the calculation, molecular docking calculations. What is the information from molecular docking result? It is first is okay. The energy is I say that the energy is the what is called. We first talk about the interactions. For example, the main interaction of erlotinib on EGFR. This is the MET 769. This is the interaction, a specific interaction for the activity of the compounds. So finally, we have make the many. I think at that moment, at that time is 10 molecules. The variation of R1, R2, and R3, and this is one of the interesting what is called results of the calculation is 1N. This R1 is ethyl, R2 and R3 also the ethyl. R1, R2, R3. So this is the important calculation, or we make the calculation here is there is the specific interaction here, and we also get another hydrogen bonding here, this one. So it means the possibility to have the activity, high activity, or increase the activity of compound. This is because of another interaction, not only with this residue, protein residue. Okay. We are also doing molecular dynamic simulation. I think it is almost software of molecular docking is also running with molecular dynamic simulation, or you can use another software to make the molecular dynamic simulation. How we use the why we need molecular dynamic simulation? It is because if there is interaction, this one, for example, with methyl, meth 98. So we can find that this is really interaction, not just come and then not the same situation. For example, this is hydrogen bond distance. If we have the simulation here and this hydrogen bonding distance, so we can say that in average, hydrogen bonding distance is about 2 angstrom. Means there is a stable, the complex is stable. This is about the molecular dynamic simulation need for molecular docking calculation. We have also another parameter like distance. This is a distance of the in hydrogen bonding calculations. The second recommended compound is R1 is H, R2 is aldehyde. This is R3 is OCH3. And we can find the interaction here that this is still have MET 769 is still here, so that we can say that the interactions is specific for the activity of the compounds, plus many residue, another calculation. Again, we running molecular dynamic simulation to check to make sure that this interaction is really interaction, not only just interact and then disappear after some picoseconds of the calculations. If we have data about interaction, so we can running what we call pharmacophore modeling of protein EGF. This is an important thing for designing the molecule from the calculation of molecular docking. For example, if we talk first one is the form hydrogen bond is in the main catalytic region, namely the amino acid residue. This one. So it means we try to make the new compounds, not make, but we can find new compounds that form the hydrogen bonding with specific amino acid residue here. The second one from this calculation, there is an additional interaction, which is or associated in this residue that so that the drug compounds they have a good activity. It is kind how to make the station of the compound that we can try to molecular docking and then give to the people who running their synthesis in the laboratory. Okay, that is the third one, and that is fourth one. So this is what we call pharmacophore modeling. After we have some what is called some points, interested point to make the decision of design. So we try to make this one. Yeah, of course, for molecular modeling, very easily to just change R1, R2, and 3 with another atoms or substituent. Yeah, but of course for synthesis, this is the problems. Yeah. Okay, but we start from here. We check R1, the more variation until 3 type, A, B, and C with the different substituents. Substituent here. Finally, we have the interaction of recommended compound EGFR. This is from molecular docking calculations. For example, we should check the hydrogen bonding. Met 769. Yeah, it is the same. There is hydrogen. So means molecule A1, A2, A3. It is possible to use. But here is hydrophobic interaction is a very quiet, so that there is additional interaction of the compound. That's why we choose A1 as a recommended compound for the activity of EGFR. Again, I don't want to give you the molecular dynamic simulation for this, but we have simulations about how stable our conformation on interaction between protein and molecule in drug. Yeah. Okay, this is the interaction recommendation compound. EGFR is another. We have class A, type A, B, and C. Wow. One B, this B1, and then C as C1. Okay. After we finish for the modeling of the compound, and we have the data that 1 or 2 compound is recommended for the synthesis. Normally, our collaborators also make the retrosynthetic analysis compounds. It is actually because I am also the synthetic in chemistry. So if we have the retrosynthetic analysis, mean that give the information to the people who are working in synthesis that it is possible to running. Otherwise, the people in synthesis just, it is a very expensive, this is a very difficult to running and so on. But we try to give the informations, yeah. Maybe we can not, the researcher in synthesis cannot choose this retrosynthesis analysis, but we can give the possibility that it is possible to use for synthesis, yeah. Of course, after that, we have the synthesis of compound. I don't want to detail for this, but of course, it is just give you the information that if we have just not recommended the compound, but we also has the synthetic route of the compounds. Okay. Yes, it is just an example. Another research on computational chemistry, especially molecular docking. This is molecular docking of the compound. This is okay. Another I think is Mario. So he liked this, and then we have also the synthesis. This is not only molecular docking calculation, but he also running the synthesis in the laboratory, so that he have the possibility to running the molecular docking calculation, but also have the working in synthesis. Yeah. Example, not only the running computational chemistry calculation, but also the doing the calculation, doing the synthesis. Okay. There's another example is only using the QSAR, but actually, what is the name? Juanita, Amy Juanita is also start from calculation with QSAR and then doing molecular docking calculations and then doing the experimental for synthesis this compounds, yeah, until they get the activity. Another example is, yeah, this is a new publications. The student is still working with a synthesis. Yeah, the first publication is about fully using the computational chemistry calculations. Yeah, okay. Another example is the same. Mutiara Ayu Iresa is also running the calculation with molecular docking calculation, and now I think this is quite not bad, but you know, they have a 6 recommended compound from computational chemistry calculation or molecular docking calculation. But until now, they have she has 2 compounds, yeah, 2 compounds already, but some difficulties in synthesis, really synthesis the compound. Yeah. Okay. Yes, thank you for your attention. I just give you the learning by doing, understanding by teaching, and designing by molecular docking. Yeah. Okay, thank you very much for your attentions. Yeah. Thank you.
All right. Thank you, Professor Harno, for delivering the talk about the drug design based on the study of the molecular docking. There are QSAR analysis, for example. And all right, we come to the Q&A session, and we have questions in the chat. Maybe Anissa Nurjana would like to ask directly to Professor Harno. Anissa, are you there?
Oh,
right.
You can.
Yeah. Otherwise,
no?
yeah. Otherwise, we can read.
Yeah, we can
question.
read from the chat. Yes. How to do ligand optimization for peptide protein docking since peptide docking can be a little bit tricky due to its highly rotation bonds. Okay, I talk about the what we call simple interaction because we have the drug compounds as normally small compounds and the protein receptor protein receptor of this. Yeah, so that the more flexibility is not only in the drug but in receptor, molecular receptor. But Anissa asked about how we use the peptide, or we can the molecular docking also not only the drug molecule or small molecule, but using also protein molecular docking. Yeah, means that big molecules. Yeah, and it is quite different. What molecular docking is the same, the technique, but how to make that the sub conformation of the big compounds is reduced. Yeah, so that we have some several methods. Like we just give you the information about the short, what we call in QMM, quantum mechanic molecular modeling. We have just give the short box simulation, small box simulation to make the possibility to reduce the flexibility of the molecules. You can still use the Autodock and so on, or another software for not the small ligands, but use the peptide protein docking. Yeah, the big what is interesting to use this is if you have the structure, the what they call the initial structure of the compound is similar to the last structure after molecular docking. Normally, we still use the XRD result of the compounds. From this part, we can make the calculation of molecular docking. So it is the technique how to. It is quite different. Quite different the technique of running with a small molecule. Yes, I'm sorry we have running in Yogyakarta, so not so. Okay, Anissa.
Right. Thank you,
We
Prof.
do another.
Yeah, another question, Prof.
Podlengga Priani. Yeah. Okay, thanks for praising, Prof. I have a question regarding how to predict the combination interaction between active compound. Whether there is the same as predicting of interaction of molecule in the compound, because sometimes, just a moment, the same as predicting, because sometimes in a drug, there are several active compounds. For example, oral drug. To maintain the stability of drug, okay. This is quite different. In molecular docking calculation, we cannot put another factor like in experimental. In molecular docking calculation, this is like, we just running that compounds is already docked, already near the active site of the compounds. We cannot give you the interaction with how the compounds through the membrane and then come to the site active of the compounds. So we cannot predict this one using the molecular docking calculations. But of course, Prof. Narstad say that we still using the experimental value for the calculations. Yeah, to know that how the interaction between ligand and receptor during the process to go to the receptors. But molecular docking is now it's not only the interaction between molecules, drug molecule, and the receptor in the same, what is called the same field, the same what they call. We don't think about the, for example, log P. If we talk about the log P, means that the log P partitions, yeah, we know that big molecules, crowded molecules, is physically is very hard to go to the membrane, to go through the membrane. And this is not include the calculation in molecular docking calculations. Yeah. Okay, this is the possible. So, for example, in oral drug, to maintain the stability of drug during the process of the drug molecule to come to a receptor is not constrained by molecular docking calculations. Yeah. Okay.
Right. Thank you, Professor Harno, and thank you, Lengga, for your questions.
Yeah.
Yeah, we have another question.
What type of energy that is calculated? Is this bond energy in ligand or non-bonded energy between the ligand and receptor? The tyrosine kinase inhibitor, sometimes inhibitor can bind covalently to the receptor. Okay. The question is about what kind of interactions predicted during the molecular docking. If we talk, take a look the energy of molecular docking, the calculation. Actually, we have some or several. May I share again the structure here so that yes. Just a moment, I have. Okay, for example, this one. This is the type of interactions. For example, this is the important thing is hydrogen bonding. Another energy is from hydrophobic interactions. Yeah, means there is not only the hydrogen bonding, but also the hydrophobic interaction include also their pi interaction and so on. If we want to look the real, I mean not the real, the every type of interaction, you can find it. Yeah, you can find it in the result of the molecular docking. Not only the hydrogen bonding here, but also all the interaction. If you take a look this, where is, yes, oops, sorry. This one is only the interaction, but here, every color here is different meaning of interactions. Yeah, this one is hydrogen, what we call conventional hydrogen bonding. And I don't exactly know, because normally there is a put here the name, the type of the every color here, pi interaction, or maybe just a van der Waals interactions. So that all interactions in molecular in interaction between ion molecule, drug molecule, and protein receptor is calculated. Yeah, of course, if you find there is no example here, but with the total energy, S means that the total for all the interaction in their molecular docking calculations. I think this is our. Oops, sorry. Yeah. It is another
Yeah, thank you,
Yeah.
Professor Harno. Yeah.
Yeah.
Another question from Pika.
This is the new one. Okay. Which interaction we like and find the molecular docking? It is the same interaction that native ligand had, or interaction between our compound and the protein does matter. Yeah, I think it's already give you the information. We have several type of interaction during the molecular docking calculation, and conventional hydrogen bonding is the important one. You know, the structure of protein is start from the hydrogen bonding, the calculation actually, yeah. And then we just know that interaction between between molecule of the drug and the protein receptor is I don't know exactly the average, but maybe more than 50% is because of the hydrogen bonding conventional. Yeah. After that, we have many interaction, like you know, if the molecule is very flexible, for example, so that we should know van der Waals interactions. Yeah. Normally, van der Waals interaction is also important for the structure, that a lot of flexibility. Yeah. Degree of freedom is high, so that we should appoint it for the van der Waals interactions. Yeah, will give us a different interaction, and some of them are not the interaction that native ligand protein complex had. Yeah, that's why I always say that supramolecular chemistry is very important for doing or for interpreting, yeah, the interactions, not just the energy itself, but the important thing is how and explain what kind of interaction in molecular docking give us the possibility to design molecules. It is very good example. For example, you need the hydrogen bonding, so need OH, yeah, OH, OH of in the molecular drug. So you make the decision is you change OH with OCH3, for example, just hydrogen to change the methyl. It is very big, the problem, because you cut it, the possibility to make the hydrogen bonding. For example, this is just example that we should know the possibility to design molecule, not only just change the substituent, but we should know about what kind of interactions needed for the have the such activity. Yeah. Okay. Thank you. I think it is. I hope that you can have the answer from our discussion. Thank you.
All right. Thank you, Professor Harno, and thank you, Pika, for your question. All right. Before we come to an end of our webinar, maybe if you still have questions for Professor Harno or maybe for Professor Nares, you still have chance to raise your hands or deliver your questions. Otherwise, maybe if participants here have some questions, maybe they can send you email, Professor Harno, if that is okay, or yeah, if participants have questions.
Oh, yeah.
So you can yes, yeah, so participants can directly keep in touch with Professor Harno or maybe with Professor Nares.
Yeah, thank you.
Right. If no more questions and no more discussion, we, on behalf of the Department of Chemistry Education, Universitas Pendidikan Indonesia, we also would like to gratefully thank to Professor Harno for your availability and your time to deliver such a great talk in this afternoon. Thank you, Professor Harno.
Welcome.
Thank you. So hopefully in the next meeting or next event, we can meet again with you, Professor Harno.
Yeah.
Yeah, so thank you very much. And also thank you for participants for attending this webinar, even though we are doing it virtually. And the attendance link and the feedback has been shared in the chat. So don't forget to fill out the form. And maybe from our department, Ibu Fitri Khairunisa, are you there? Maybe you can have words to say, Ibu Fitri? All right. Ibu Fitri was actually here, but yeah, maybe she has occasion. All right. Once again, I would like to say thank you to Professor Nares and then Professor Harno. Thank you for your time and for your availability. Thank you for your lectures, and also thank you everyone. We would like to remind you again to fill out the attendance and feedback form that has been shared in the chat room. And I would like also to remind you that in next month, November, Professor Nares will still give the lecture. It will be about polyfunctional compounds, especially the reactions of the dicarbonyl compounds. So make sure you already mark your calendar and then attend the class. But it's now 3 p.m. in Bandung, Indonesia. Thank you. Thank you all. So see you again in the next event or in the next webinar. Thank you, Professor Harno. Thank you, Professor Nares.
Okay, welcome.
Bye.
Thank you. See you again.
Yeah.
Yeah, or for everyone, please fill out the attendance and then the feedback. And don't forget to attend next month's Visiting Professor lecture series. Yeah, here we show you the attendance link. Terima kasih semuanya.